That's it. Also, can you read it to the corner?
A very happy afternoon, students. Yeah, welcome back to RGR Academy. So, to the biology session. I hope you're all safe and sound at home. So, let us begin with today's chapter, that is plant physiology. So, we started with the mineral nutrition in the last class. I hope you remember we had discussed about the macronutrients as well as the micronutrients. There we discussed about the essential and non-essential nutrients. As far as the plant physiology is concerned, right? There are a few chapters that I told you already, like we can give priority for these chapters, that is photosynthesis, respiration, and coming to this nutrition and mineral nutrition, where like we can give importance for micronutrients as well as for the macronutrients. Also, the nitrogen cycle, the nitrogen fixation, symbiotic and asymbiotic organisms. These are like bit of areas that we need to show an interest and so that we can expect the questions from this part also. As far as the mineral nutrition is concerned, this is a continuation. The chapter, the nitrogen metabolism, is a continuation of the mineral nutrition, that same chapter. Hopefully today we complete this chapter. We discussed in this particular session, we discussed about the nitrogen metabolism, the nitrogen cycle, and all the stages which involve in the nitrogen cycle that we're going to discuss in detail. As far as this particular chapter is concerned, so what are the areas that you can expect questions? You can expect questions. I told you like the the macronutrients, the trace elements that you can expect, the respiratory quotient that you can expect a particular area, and also the deficiency. We discussed in the last class about the deficiency, right? So in that particular area, we can get a question. So the deficiencies like molybdenum, boron, zinc, iron, ferric, nitrogen, sulfur, magnesium, manganese. These are the areas that we can get a question. So guys, we keep our fingers crossed, and I hope you consolidated all the points that we discussed in the previous class, where we discussed about the macro elements as well as the micro elements, the functions of these elements, and also we discussed about the symptoms of these elements. That's what actually we discussed so far. I hope you're doing like all the necessary preparations at home. You're doing a practice. You're going through the test papers. So only the practice that can make you perfect. So my humble suggestions is like of course you're listening to the class. Of course it's good, but same time have enough practice. Have enough practice. Go through all the test papers. Make use of all the resources that we're giving it for you. So make use of this and hope for a high marks as you expected, right? So this chapter, in this particular chapter, today we'll start with the nitrogen metabolism. So we'll start about the nitrogen metabolism. Okay. So coming to the nitrogen metabolism, what are the things that we're going to discuss in this particular chapter? The nitrogen metabolism. We'll discuss about the nitrogen cycle. The nitrogen cycle, where different parts of the nitrogen cycle, that is ammonification, the nitrogen fixation, root nodule formation, then nitrification, denitrification, and also the fate of ammonia that we're going to discuss in the chapter, and the other mode of nutrition, the heterotrophic mode of nutrition also that we're going to discuss in this particular chapter. So on the whole, this chapter, the mineral nutrition and the root no, I mean the nitrogen fixation, you can get three questions. Okay, three questions that you can, one definite question that you can get from the elements, that is micronutrients or from the macronutrients. The other questions like you can get from the enzymes, especially the nitrogenase enzyme, that area you can get a question, or the nitrogen fixation, or from the nitrogen cycle you can get a question. So let us begin with today's session. So I hope you already revised with this particular chapter using a textbook. So it will be easy for us to have the all the necessary information so that we can gather it and we try to consolidate and we try to complete this chapter by today. If time permits, we'll start with the plant growth regulators, PGR, we call it as plant growth regulators. We discussed about the hormones today, like auxins, gibberellins, cytokinins, abscisic acid, all these hormones that we can discuss today if time permits, or else we'll just stop with this particular chapter itself. So let us begin with this particular chapter. The today chapter is nitrogen metabolism. Coming to nitrogen metabolism, So what is actually the nitrogen metabolism is all about? So because nitrogen is actually the important component actually for the plants are concerned. It's also for indirectly we are also depends on the nitrogen because we depends on the plants where the plants requires the nitrogen for its metabolism because plants cannot fix directly like the nitrogen. It has to be converted to the nitrate or it has to be converted to the ammonium ions. Then it will be like taken by the plants actually. So the complete process that we're going to discuss in this chapter by nitrogen fixation because the nitrogen metabolism is a component of you could see like in the form of amino acid in the form of protein like in the form of vitamins these for all these like components for this it requires nitrogen as I told you like in atmosphere we could see the majority the component that is the nitrogen which comprises of the 78 percentage and then followed by the oxygen of 20 percentage and the carbon dioxide which is of 0.038 percentage. So how the abundant this particular nitrogen is going to get fixed because this nitrogen which present in the atmosphere we call it as a denitrogen. It's not the routine nitrogen we call it as denitrogen and this denitrogen has to be fixed first actually. This process called as nitrogen fixation, right? So this nitrogen fixation which happens by means of the microorganisms. There are various ways how this nitrogen getting produced by means of industrial, by means of lightning, by means of thunder, by means of biological mode. That these are all the classifications that we're going to discuss as well in this particular chapter. Fine. So let us begin with this part. First, we'll start with the nitrogen fixation. All right. So coming to the nitrogen metabolism, So as I already told you, nitrogen is important component. It's an important component for amino acid, right? It's important component for amino acid. It's for the vitamins. For all this, like the nitrogen is quite seems to be essential. So, in what form this nitrogen is present in the atmosphere? This is present in the atmosphere in the form of dinitrogen. This dinitrogen, which present in the atmosphere, which cannot be directly taken up by the plants that I already told you, it needs to be absorbed. It needs to be converted to some other form and absorbable form by the plants, so it can be utilized by the plants. So the process that we discussed, like ammonification, nitrification, denitrification, like how assimilation of this nitrogen is all the things that we're going to discuss in this part. Okay, the conversion of inert nitrogen to utilize the compounds of nitrogen like nitrate is termed as nitrogen fixation. So where we discussed about like biological as well as the abiological. So nitrogen fixation, the biological terms also we discussed. There is a specific term that we'll give for the biological nitrogen fixation. What is the term that we usually give for the biological nitrogen fixation? The specific term compared to the abiological mode. The specific term that we'll give for the biological nitrogen fixation. Can someone tell me what is the term that we give for the biological nitrogen fixation? Fine. So coming to this process, the nitrogen's metabolism, in the atmosphere you could see this is actually present in the form of denitrogen. We move on to the next process here, where in the atmosphere we have the form called denitrogen. This denitrogen has to be converted. This denitrogen has to be converted to the other forms like in the soil. In soil, what are the forms that we could see? You could see in the form of nitrate. The form of nitrate. You could see in the form of ammonium. You could see in the form of ammonium ions. So as the plants cannot take directly this type of nitrogen, dinitrogen which is present in the atmosphere, this has to be converted into the form of nitrate, or it has to be converted into the form of ammonium ions, that is NH4. So the supply of nitrogen is maintained by means of the cycle that we call it as a nitrogen cycle. We call this cycle as a nitrogen cycle. So now we move on to the parts of the nitrogen cycle. When come to the parts of the nitrogen cycle, we discuss like ammonification. So what are the parts that we discuss? Ammonification, denitrification, ammonification, nitrification, then assimilation of ammonia that we'll discuss, then denitrification. These are the main stages that you could see in the, and also nitrate assimilation also that we'll discuss under each segment. So ammonification, we have the nitrification, we have denitrifications, all these things that we're going to discuss, and also we have the nitrogen fixation. We're going to discuss in this particular chapter. Now we'll begin with the nitrogen fixation. So we'll begin with the nitrogen fixation. So how the nitrogen fixation takes place? The nitrogen, actually the dinitrogen, which cannot be fixed directly by the plants and the animal. So the atmospheric nitrogen, which has to be converted to ammonia, this process we call it as a nitrogen fixation. So the nitrogen fixation where the atmospheric nitrogen where it is getting converted to ammonia, right? This process we call it as nitrogen fixation. So what's happening in the nitrogen fixation? This nitrogen fixation usually of two types. One is abiotic and other one is the biotic. Coming to the abiotic forms, we could see the abiotic where the atmospheric way of like fixing the nitrogen and industrial nitrogen fixation. Coming to the atmospheric fixation of nitrogen, we are thunder, we are electric discharges, which requires high temperature, which requires high energy for this, right? So, and the industrial mode of like fixing a nitrogen. Coming to the biotic form of fixing nitrogen, the biological fixing form of fixing nitrogen, we are with the help of the microorganisms like the bacteria or cyanobacteria, that is blue-green algae, this nitrogen can be fixed. This can be classified again symbiotic and asymbiotic. You know the difference between the symbiotic and asymbiotic. If we have a mutual relationship that you could see, right, between the host and the bacteria, then definitely we call it as a symbiotic relationship, mutual relationship that you could see. But in case of the asymbiotic, it won't be having a mutual relationship, it's free living. This asymbiotic usually it's of free living organisms usually present in the soil, but somehow it can help even the plants to perform this particular plants can take up fix the nitrogen. So this can also happen by means of free living microorganisms like asymbiotic organisms. Symbiotic organisms, especially you could see in case of rhizobium, in case of frankia, the examples where you could see the symbiotic organisms, it should be the part of the plant body itself, the plant root itself. You want to study the root nodule formation. These particular bacterial species like rhizobium, which gets localized in the root nodule itself, where this facilitates the fixation of nitrogen, and this nitrogen which will be getting supplied to the plants, and the plants will be benefited, and both organisms, both the bacteria as well as the plants, both will be benefited by means of the symbiotic relation. It's a mutual relationship actually, because there is no harm, right? They're both the partners, where the plants as well as the bacterial species both are going to get benefited. This you have studied in the 12th standard portions like negative interaction and the positive interaction. You have studied the various types of interaction like mutualism, commensalism, amensalism, predation, parasitism. These are different types of interaction that you would have studied. But out of which here you could see the interaction between the plants and the bacterial species by means of the root nodule formation is a kind of symbiotic relationship where both the partners, where both the population where is going to get benefited. It means where the root node, the leguminous plants and also the bacterial species or the cyanobacteria, both are going to get benefited. And for your kind of information, this cyanobacteria and this root node or the rhizobium, usually it is asymbiotic in the soil. When the time comes where the nitrogen fixation, right, the nitrogen fixation comes, that time only what happens, this rhizobium get inside and it forms infection thread and form root nodules. This will become a symbiotic organism. But usually the rhizobium bacteria, it's an aerobic bacteria. You don't be having any sort of mutual relationship in the beginning. When the mechanism called as root nodule formation, at that point of time only the rhizobium and the leguminous plants start with the relationship where it go for the mutualistic relationship. Fine. So yes, Kamadeni, you asked what is a doubt. So this is how actually the symbiotic, but when we usually what we think is like symbiotic organisms always will be having a close proximity. It will be having a close proximity with the root nodules, right? But to be frank, this symbiotic organisms, the rhizobium is concerned. Usually it go through a asymbiotic mode of adaptations. But later point of time, what will happen when it go with the process of fixing a nitrogen root nodule formation? This goes with the symbiotic relationship. But in the the starting phase, you could see the rhizobium, or you could see it's also like the other microorganisms. Usually it is like asymbiotic. Okay, so as well as symbiotic root nodules and stem nodules, sometimes you could see the leaf nodules also that we're going to discuss. So we're going to discuss the root nodules and the stem nodules. Fine. Coming to the symbiotic, we have the root nodule formation and we have the stem nodule formation. So this is the classification of the nitrogen, the fixation, the classification of nitrogen fixation. Now what we're going to discuss is, we discussed first about the abiotic that we discussed about the atmospheric, and we discussed about industrial sector. As far as industrial part of the thing is concerned, I told you the industrial sort of fixing nitrogen, which requires high temperature, and it requires high temperature first, and also this requires high amount of energy. It requires more amount of energy. You could have seen in most of the industries where you could see in this case where this will be going with the help of like the urease enzymes, where what happens, like the compounds, like for example you could see like NH2, right? This NH2, this is like NH2, this is getting combined and it forms 2NH3. This by means of the enzyme called as urease enzyme. So usually the help of the urease enzyme, with the help of high temperature and high energy, these compounds can be getting converted to the ammonia with the liberation of carbon dioxide. This type of reactions that you could see in case of industrial nitrogen fixation. Okay, industrial nitrogen fixation. So excuse me, this is not chemistry, this is biology. Okay, fine. So where the industrial type of nitrogen fixation is, which requires high temperature and high energy. Coming to the atmospheric form of fixing, the abiotic mode of fixing nitrogen by means of thunder, by means of electric discharge. So what happens like atmospheric fixation of nitrogen, that we'll discuss, and we move on to the back. So abiotic. First we'll start with the abiotic. Abiotic, we already discussed about industrial nitrogen fixation. Then we have atmospheric. Mode of fixing nitrogen. Once we complete this part, we move on to the next area. So coming to the atmospheric fixing nitrogen, where the nitrogen is combined with the oxygen and it forms 2NO and plus it forms H2O, right? And this they call it as nitrous oxide. And then this nitrous oxide, which combines with oxygen, and this forms 2NO2. This is nitrogen peroxide. Which forms. This is nitro oxide. So these reactions which takes place by means of, you could see, the lightning or by means of electric discharge. This happens by means of lightning. This reaction takes place by means of lightning. This happens by means of electric discharge. This nitrous oxide which gets converted, right? This process, we call it as a process called ozonization. By means of ozonization, yes, it is ozonization. This is called as ozonization. By means of this ozonization, where this nitrous oxide which combines with 2 molecules of oxygen and just it forms like nitrogen peroxide. So what happens to this nitrogen peroxide? Again, the 2NO which combines with 3 molecules of oxygen and then it converts to 2NO5. So what happens now? 2NO plus 3 molecules of oxygen, which gives rise to N2O5. So these reactions which usually takes place, this is actually ozonization process which takes place. This comes into the category of atmospheric fixation of nitrogen. So what is the fate where what you're getting is actually the nitrous oxide, then nitrogen peroxide. Then what happens? This nitrous oxide, this nitrogen peroxide, this will combine with the water. Then what happens? Like 2NO2, which combines with water. So what you'll be getting, right? So if this 2NO2 which combines with water, then you'll get 2HNO3. So this will get 2HNO3. That is like nitric acid. This forms nitric acid. This will be getting the form of rain actually. In the form of rain, the rain which will be carrying this nitric acid. So this nitric acid which is getting penetrated to the soil again, this will be increasing the acidity of the soil as well. So this reaction that you should be very careful with the atmospheric form of nitrogen fixation. This occurs by means of lightning or by means of electric discharge. So what you'll be getting is nitrous oxide and you get nitrogen peroxide. And nitrogen peroxide, which by means of ozonation process, you get N2O5. This N2O5 which combines with the water. when combines with the water, then what will happen? You get 2HNO3. So what happens, like, this 2NO2 will combines with the water, will get a 2HNO3. Okay, fine. So this is how actually you describe the atmospheric form of fixing the nitrogen. So these two things comes under the abiotic forms, abiotic form of fixing the nitrogen. Okay. Now we move on to the next process, that is biological form of fixing nitrogen. This is like abiotic. We move on to the biological type of nitrogen fixations. Coming to the abiotic form of nitrogen fixation, here you could see in this case, please, you could see in this case biotic form of nitrogen fixation. The biotic type of nitrogen fixation with help of microorganisms, this we call it as diazotrophy. The biological nitrogen fixation we call it as diazotrophy. So the other name for the biological nitrogen fixation is that we call it as diazotrophy. Okay, this biological nitrogen fixation which takes place by means of organisms. Organisms you take by means of the bacteria, or you take by means of the cyanobacteria. So this diazotrophy, that is biological nitrogen fixation, can take place by means of bacteria, or it takes place by means of blue-green algae, that is by means of cyanobacteria. Now this bacteria again classified into symbiotic mode and asymbiotic. Again the blue-green algae, which again classified into symbiotic and asymbiotic. So as already told you the difference between symbiotic and asymbiotic. So for your kind of information, so just as you requested, I just recap what we discussed previously. This is about the atmospheric. We discussed about the abiotic nitrogen fixation. We discussed about industrial mode of nitrogen fixation, which requires like more amount of energy, and it requires more high pressure, and it requires high energy and high temperature. Coming to this for atmospheric form of fixing nitrogen, where the nitrogen combines with oxygen and form nitrous oxide, with this by means of lightning, and this nitrous oxide combines with the 2O, then it gives 2NO2, then it is nitrogen peroxide. This is the electric discharge. Then 2NO plus 3O, which gives rise to N2O5. This process is called as ozonization. So now what happens? The nitrous oxide, peroxide, and which reacts with the water. Then what happens? Due to this, what happens? Nitrous and the nitric acid are formed. So then what happens? Like 2NO2, which combines with H2O, which gives rise to HNO2 and HNO3. So this N2O5, which combines with the water molecules, this gives rise to 2HNO3. Then this we call it as a nitric acid. So this is what actually you could see in this NO2. This is NO2. This is NO2. Okay, fine. So we move on to the next part, that is the symbiotic and the asymbiotic mode of nutrition. So coming to the symbiotic asymbiotic mode of nutrition, we have symbiotic. We discuss like few organisms. From this area, you get a question. So they give the examples, options. They'll ask you to identify which is the symbiotic and which is the asymbiotic mode of nutrition that you discuss. So first you'll see the free living type of nitrogen fixing. So free living is actually the asymbiotic mode of organism. This may be anaerobes. Anaerobes means completely the lack of oxygen. Facultative anaerobes means maybe and may not be. Microaerobics means only it requires little amount of oxygen. Aerobes means completely it requires oxygen. Right. Then photosynthetic bacteria. Then we have the cyanobacteria. These are all the types of different groups of bacteria. Right. This we call it as the free living nitrogen fixing organisms. Coming to the symbiotic systems, like symbiotic, you could see Rhizobium legume association. This again we like classified as the fast growers, and it's also be called as slow growers. And Rhizobium can also have the association like with the non-leguminous plants also. You don't think like legumes, Rhizobium, which have the relationship only with the leguminous plants, but also it's having the non-leguminous plants, but usually these are all slow growers. And associated symbionts, then we have Frankia, which comes under the Actinomycetes family. So in the bacteria, this comes under Actinomycetes, the Frankia, which comes under the Actinomycetes family. Then the cyanobacterial associations, we have angiosperm, gymnosperm, pteridophytes, bryophytes. Then we have lichens. This lichens, right? Okay, this lichens. Okay, this lichens, what is a lichens, right? So lichens is the symbiotic relationship. You have studied about the mycorrhizae and the lichens. So what do you mean by the lichens here? Lichens is a symbiotic relationship between the algae and the fungi, right? Between algae and the fungi. Okay. So since I couldn't zoom it now, that's the reason. Like if you want, I'll send the image for you, right? Because I phrased it already, so now I cannot zoom it and show you. Okay. Anyway, I'll just dictate whatever thing that I'm reading. I'm just dictating it for you if it is not visible for you. So the symbiotic system, rhizobium legume association, rhizobium non-legume association, associated symbionts. Then we have the frankia here. Frankia, then cyanobacterial associations, angiosperm, gymnosperm, pteridophytes, bryophytes, and lichens. Lichens is a symbiotic relationship between algae and the fungi, algae and the fungi. The same way you would have studied like mycorrhizae. Mycorrhizae, what do you mean by mycorrhizae? You study like ectomycorrhizae, endomycorrhizae, eckendomycorrhizae, vesicular arc-vascular mycorrhizae. Sure, I'll send this image in the group. Okay, sure, I'll do that for you. So the same way, this is also examples where you could see the lichens, the algae and the fungi, which goes with the symbiotic relationship. Look into the examples from into the free-living nitrogen-fixing organisms, anaerobes. Usually the Clostridium is a bacteria which requires like anaerobic conditions. So Clostridium. Facultative anaerobes, we could see Klebsiella and Enterobacter species. Microaerobes, Azospirillum, Aquaspirillum, Arthrobacter. Aerobes, Azotobacter. So Azotobacter, Dexia. Photosynthetic bacteria, Rhodosudomonas, Rhodospirillum, and we have a Chromatium. Okay. Then cyanobacteria, we have Anabina and Nostoc. So these are all which comes under the free living. It means, what do you mean by free living? It is asymbiotic, which don't have any close relationship with the leguminous plants, because it's like living in the soil. They don't have any sort of commitment, they don't have any sort of interaction or direct relationship with the roots of the plants. So this is what actually it means by free living organisms. Fine. So coming to the symbiotic system usually mostly with the rhizobium species and with the frankial species, and also we have with the cyanobacterial association that I discussed, like few examples with the angiosperm, gymnosperms and all. The frankia will see usually with the alnus, casuarina, myrica, M-Y-R-I-C-K, myrica. So this is myrica, M-Y-R-I-C-K. Guys, do not worry about the font size. I just send this image to the group. Okay. Then Parapsonia, then Arachis, Glycine, Virginia. These are the slow growers. Fast growers, Pisum, Trifolium, and Vicia. So these are the examples from this particular area, right? So why I'm showing this slide, we're giving examples is that this is from this area you get a question from the symbiotic. They give 4 options. They'll ask you to identify this following as a symbiotic or the following is a asymbiotic, right? So from this area, so from this, like you get this type of question. Okay, this type of questions that you can get from this area. Okay, so you clear with this part, that is with the symbiotic and asymbiotic mode of nutrition. Okay, fine, we're done with this part. Then we move to the next part, that is biological nitrogen fixation. So by the bacterial species we discussed, and the other form that also we discussed, that is symbiotic form, that we'll discuss. So now we move on to the next form. Fine. We move to this part by bacteria. First we'll start with the bacteria. By bacteria. Okay. By bacteria, I told you it's like free living, and we are symbiotic. Right. Coming to free living, this can fix 10 to 25 kg per nitrogen per hectare per annum. Okay, so coming to free living form, I told you already about aerobic, anaerobic, and also we discussed about facultative, aerobic, anaerobic, and we discussed about facultative organisms, that also we discussed. Okay, fine. So aerobic, anaerobic, and we have the facultative organisms. Coming to the aerobics, examples that they ask for the aerobic, we have Azotobacter. The few examples that you can remember: Azotobacter, Beijeringia. So, Azotobacter and Beijeringia that you can get. So these Azotobacters and Beijeringia, these are usually saprophytic in nature. Saprophytic. Coming to the anaerobic, and on this also you could see, like, aerobic organisms. We have the Azospirillum also. We have the Azospirillum. Okay, Azospirillum also we can get it. So Azospirillum is a chemoautotroph. This is Azospirillum which comes under chemoautotroph. Like this type of questions also that you can get, like which among the following which is aerobic. So what is the mode of nutrition? So here Azospirillum is a chemoautotrophic, whereas like Azotobacter and Beijeringia, these two which comes under saprophytic. These are all saprophytic organisms. Fine. Okay. Now, then we discussed about the anaerobic mode of organism, D-sulfovibrio. Examples we take D-sulfovibrio. Examples for anaerobic, we could say D-sulfovibrio. Then we have the vibrio and we have a Clostridium. We have vibrio and Clostridium species. Vibrio and a Clostridium species. This comes under anaerobic. Then comes the facultative. Facultative usually Bacillus species, and we have Klebsiella. Okay. So this is how we can classify the free living organisms, and how much of the nitrogen it can fix is 20 to 25 kg per nitrogen per hectare per annum. So this is what the range this can fix it. Okay. Coming to symbiotic form. Symbiotic form, we have the Rhizobium, Frankia, Xanthomonas. We have a symbiotic Rhizobium, Frankia. These are the two main organisms that you can categorize under the symbiotic organism. The Rhizobium and we have a Frankia. And we have Xanthomonas and also we have Mycobacterium. Also you can include Xanthomonas and Mycobacterium. So these are some of the species which can fix atmospheric nitrogen. Okay. This can fix atmospheric nitrogen. Fine. So the Rhizobium, Frankia, then Xanthomonas, then the Mycobacterium. These are examples for symbiotic. And Rhizobium you take, this can form like stem nodules, and also this can also like which leads to the root nodules. For example, you take Sesbania. Sesbania is example. I just give example for Sesbania, which is responsible for formation of root nodules. So you cannot expect the root nodules only in the stem. Root root, but also you could see the root nodules in the stem part also. Sesbania you could see in case of the stem nodules. So the Sesbania you could see in case of stem. The stem nodule it forms the Sesbania. Coming to the next part, the root nodules, you could see Rostrata. Rostrata is a species, Rostrata, which have the ability to form root nodules. If the question comes like which of the following which has the ability to form stem nodules, Sesbania. Coming to the root nodules, Rostrata. Rostrata is actually the root nodules. Fine. Coming to the Frankia. Frankia, what are the types of plants that usually you could see in case of Cycas, the Halnus? Then Xanthomonas. Xanthomonas you could see in the leaves. Xanthomonas you could see in the leaves of Rubiaceae. So these are the areas, the nodules you could see. If the question comes like, if the stem nodules, Sesbania, root nodules, you take Rostrata, the leaves of Rubiaceae, then you go with the Xanthomonas. So these are all like, areas like, if the question comes which among the following which is responsible for the stem nodule, that is Sesbania, then root nodules, Rostrata, then the leaves of Rubiaceae, it is the Xanthomonas. Okay, fine. This is how actually, this is how we can categorize the bacteria, like, the free living, and we classify it into the symbiotic mode of classifications. So there are a few examples that I can give along with this. You could see there are a few cases where the Rhizobium and Frankia, I told you, the Rhizobium and Frankia usually it will be in the form of asymbiotic mode of nutrition, the asymbiotic mode of nutrition. But when it comes to the association with the plants like leguminous plants, then you could see like this actinomycetes like the frankia and the rhizobium, which will become the symbiotic relationship. It will become like symbiotic association. Until and unless this won't be having any relationship, because the rhizobium and the frankia usually be in the free living form. Only at the time of root nodal formation, this rhizobium and the frankia which will be going with a symbiotic relationship. Okay, so xanthomonas, which forms the leaf nodules, especially the leaves of the family called as rubiaceae. Okay, then rostrata for the root nodules, and sesbania is for the stem nodule. Okay, fine. So we're done with the classification of the bacteria by free living as well as by means of the symbiotic. And this image, which denotes this, actually the image of the leguminous plants, and this is how actually the root nodules look like. In this root nodules only, you could see in the leguminous plants the rhizobium bacteria with help of the nitrogenase enzyme. And nitrogenase enzyme, you could see the organisms proliferate and it can fix the nitrogen. Okay, facultative I told you already, bacillus and Klebsiella. In this case, I told you for free living, facultative free living organisms, that you could see bacillus and Klebsiella. Fine. So this is the classifications, that is like biological nitrogen fixation by the bacteria. Now we're going to discuss about the cyanobacteria. Okay, we discussed about the cyanobacteria. So let us quickly recap. By bacteria, it can be classified into free living and symbiotic. Coming to free living, it may be aerobic, anaerobic, or facultative. Coming to aerobic, Acidobacter, Bergerincia, these two are saprophytic. Azospirillum, it's a chemoautotroph. Coming to the anaerobic bacteria, D-sulfovibrio, Clostridium. Facultative, Bacillus, Klebsiella. Symbiotic organisms, we could see the Rhizobium, Frankia, Xanthomonas, Mycobacteria, in which you could see Sesbania, which is responsible for stem nodal formation, and Rostrata, which is for the root nodal formation. Xanthomonas, we could see in the leaves. Okay, this we have done. Okay, next we move on to the cyanobacteria. So we are in symbiotic relationship. So second part we're going to discuss by cyanobacteria. The cyanobacteria can be also called as blue-green algae. Cyanobacteria. This is also a kind of symbiotic relationship. So first we're done with the bacteria. Now we discuss about the cyanobacteria. This cyanobacteria can also be classified into free living, and it is symbiotic. This can be classified as like in case of bacteria. This can also be classified as free living and the cyanobacteria. Coming to free living form, this can fix like 20 to 30 kg of the nitrogen per hectare. So this comparatively more than that of the bacteria that we discussed before. The cyanobacteria can fix the nitrogen up to 20 to 30 kg of nitrogen per hectare. Example, you could see free living Anabaena, nostoc. Then we have Eulothrix. These are a little bit of examples that you could see in case of free living. And also Lyngbia. We have Lyngbia. Then Cylindrospermum. CY. This is CY. Cylindrospermum. Okay. Lyngbia. So this cylindrospermum, which you could see in association with the maize plant, maize and the sugarcane. That you could see cylindrospermum. Then allostria is also a kind of allostria, which is having a relationship with the rice actually, but it's not a symbiotic relationship, right? It is for the rice, allostria. Then coming to the symbiotic mode of nutrition, where you could see Anabaena. Example, here you could see Anabaena and also nostoc. These are the relationship that you could see even with the lichens. You could see the relationship with the lichens, Anabaena, nostoc. Then you could see in case of the cycas roots. You could see the cycas roots. You have the relationship with the cycas roots, and you do have a special structure as far as symbiotic organism is concerned. Which have a specialized structure is called as heterocyst, which we call it as a heterocyst. So heterocyst, for you take the algal species, which has got a special type of cell. The special type of cell, it's like triple layer cell that you could see. The triple layer cell you could see, and this type of cell which we call it as an heterocyst. And this heterocyst, what is the main function? Can someone tell me what is the main function? What is the main function of this heterocyst? The thing that we are discussing about the nitrogen fixation, that is nitrogen metabolism that we are discussing. Fine. So coming to this part where the symbiotic form in this particular algal species, we have the normal cells, and we have special type of cells that we could see. This type of cells we call it as an heterocyst. Okay, heterocyst. And this heterocyst, what is actually main function of the heterocyst? This heterocyst which use the enzyme. Can you tell me what is the name of the enzyme that we use, which is responsible for the nitrogen fixation? Can someone tell me what is the name of the enzyme? Name of the enzyme that fix the atmospheric nitrogen. So the enzyme which is present in the heterocyst. So the enzyme is nitrogenase enzyme. So this nitrogenase enzyme usually will be inactivated in the presence of oxygen. It's very sensitive to oxygen. So this heterocyst which consists of special enzyme, this enzyme we call it as a nitrogenase enzyme. Nitrogenase enzyme. Right. So usually the nitrogenase will be functioning in the absence of oxygen. That is very important. Nitrogenase which doesn't require the oxygen, and this heterocyst which maintains the micro anaerobic environment so that the nitrogenase enzyme can function effectively in this particular area, and this maintains the anaerobic state. Who is maintaining the anaerobic state here? This heterocyst is maintaining the anaerobic state, where you could see the nitrogenase enzyme which is present, and this nitrogenase enzyme which performs important role in fixing the atmospheric nitrogen. So this is what we could see in certain cases where the algal species which has got the special type of cell, we call it as heterocyst in some of the algal species. In other cases, when you talk about the root nodule formation, like rhizobium and all these cases, we'll discuss about the leg hemoglobin, how it forms with the bacteroids, because it acts like a oxygen scavengers. The leg hemoglobin which acts like a oxygen scavenger, that we'll discuss when we come to the root nodule formation. But in case of algal species, you could see the special type of cells. You could see this cells we call it as heterocyst. This consists of the nitrogenase enzyme. This nitrogenase enzyme which gives micro anaerobic environment, because nitrogenase enzyme will not be activated under aerobic state. So anaerobic state has to be maintained. So we'll discuss about the leg hemoglobin also. Okay. Example, what is that name? Fourth example. The fourth example here. Anabaena, nostoc, lichens, and this is cycas plants, cycas root. So cycas root, and here you could see Lyngbya. This is Lyngbya, Lyngbya, Lyngbya. Then cylindrospermum, this is CY, cylindrospermum. You could see in association with the maize and the sugarcane. So this categorized the cyanobacteria. This can be classified as free living and symbiotic. Okay. So exam names. Can you tell me what you mean by the exam names? What example names you're asking for? This is like Anabaena, nostoc, lichens, cycas root, and here heterocyst I mentioned. Once again, repeat examples. Anabaena, A-N-A-B-E-N-A, Anabaena, nostoc, then Eulothrix. This is Eulothrix. Okay, examples for free living, asymbiotic, Anabaena, nostoc, and Eulothrix. Then Lyngbya and cylindrospermum. Okay, so we're done with the first part, that is the classification of the bacterium, that is like symbiotic, asymbiotic. The same way it is differentiated cyanobacteria, that is free living and the symbiotic. Now we move to the next part. Next part that we're going to discuss is about, we come to the symbiotic nitrogen fixation. Come to symbiotic nitrogen fixation, the first thing that we're going to discuss is root nodule formation. Now we'll start with the symbiotic nitrogen fixation. Symbiotic nitrogen fixation. Okay, as we discussed already, how the symbiotic relationship takes place. Okay, this occurs by means of, because we discussed about the rhizobium, the bacteria, right? So if we start discussing about the rhizobium bacteria which is responsible for root nodule formation. Rhizobium bacteria, the first thing that we're going to discuss about the root nodule formation. This image which represents root nodule So I'll explain what is actually the series of stages which involve the root nodule formation. This is the first stage of the nitrogen fixation. For this, the root nodule formation, right? It requires an enzyme. This is a symbiotic bacteria that you could see, the Rhizobium. Can someone tell me like Rhizobium is of course, symbiotic. Now, Rhizobium is aerobic or anaerobic? Rhizobium is aerobic or anaerobic? Can someone tell me Rhizobium, this bacteria is aerobic or anaerobic? Okay, and Rhizobium is a gram positive or gram negative. So if you perform the gram staining for the Rhizobium, so how it look like? It look like purple color or it look like pink color. So coming to this Rhizobium is aerobic. Rhizobium is aerobic in nature, right? Coming to the next part, Rhizobium, when you do the gram staining, this looks like a gram negative. This is a gram negative. Okay, gram negative bacteria. But at the time of nitrogen fixation, this Rhizobium bacteria that is aerobic will be converted to anaerobic. So it is basically aerobic bacteria. But during the nitrogen fixation, right? Nitrogen fixation, it acts like anaerobic. It acts like anaerobic. Fine. So it is a gram negative bacteria when you perform the gram staining. So with this, we're going to discuss like with the help of this Rhizobium bacteria how so I may know what is the comments that you're posting there on that side. That Kamadenu. Stop unnecessary comments because the live class that's going on. Stop unnecessary comments. Stop unnecessary comments, I said. It's a live chat, it's going on. Stop unnecessary comments. Guys, I just place you on, mute for a while. I'll just come back. Hold a sec. Welcome back, guys. So, guys, understand this is purely the live sessions going on. So, I encourage your students, you ask or you just, ask any sort of doubts which is related to the subject. This academic sessions going on. Please refrain yourself, in discussing and like posting things which is not good for you and also for others. Okay, I hope you guys can understand. Okay, please, because completely it's a live chat is going on. So, please understand. Fine. So we'll discuss with the root nodule formation. The root nodule formation start talking about the first thing that we started with the aerobic bacteria. The aerobic bacteria, when the aerobic bacteria like this will be converted to anaerobic form. Anaerobic form when means when the time of nitrogen fixation. Nitrogen fixation this will turns to be an anaerobic. Why the reason? Because the nitrogenase enzyme. So nitrogenase enzyme it will be activated only in the anaerobic condition. In the anaerobic condition only it will be activated. So that time the Rhizobium will not be like aerobic, it will be anaerobic. This is point number one. Point number two, at the time of nitrogen fixation only this particular bacteria Rhizobium will be acting as what? Will be acting as a the symbiotic organisms. But apart from this particular process, it is like free living organisms that you could see it's freely residing in the soil itself. Okay, fine. So we're done with this part. Now we move to the first part of the nitrogen fixation. Coming to the first part of the nitrogen fixation, So what is going to occur in this diagrammatic illustration, which shows like what are the reactions which is going to take place as far as leguminous plants are concerned. So first part in the root nodule formation. So this is the root surface we're talking about. I mean the root of the leguminous plants. This is the first stage, the root of the leguminous plant. And here you could see the bacteria. This is the Rhizobium bacteria. This is the bacteria we're talking about. So just like that, the bacteria will not be like come and attach with the root hair of the leguminous plants. For that, what should happen is this root of this particular the leguminous plants which produce some sort of sugars, and this sugars which automatically which attracts the bacteria which present in the sugar molecules which produced by the root, which attracts the bacteria which present in actually the bacteria is Rhizobium which present as of now in the free living form. Now what happens after this root which starts secreting the sugar substance, right? Then this bacteria will be greatly attracted towards this particular root. Then what happens like then the bacteria which can come and get adhere onto the surface of the root, and then what happens like the first form where infection occurs and thread-like structure which forms once after the bacteria is getting attracted towards the root of the leguminous plants. Now this thread is called as infection thread. The infection thread occurs. Then now what happens? The bacteria started creeping inside this root hair and started creeping inside the thread formation, that we call it as an infection thread. Okay, yes. Usually these sugars are phenolics in nature. These sugars are basically phenolics. Yes. So this infection thread, by means of this Rhizobium, this bacteria will start penetrating inside. So this root emit the channel signals that attracts the Rhizobium bacteria. How this getting attracted? By means of the sugar, by means of the sugars. Then it emits the signal that stimulates the root hairs to elongate and form an infection. So I told you this forms an infection thread, and then what happens? This started invaginating. The Rhizobium bacteria started invaginating through the plasma membrane. Now, this is what actually the root curl-like appearance. Now, after the root infection thread occurs, the thread the root will starts forming a curl-like structure. So this curling which occurs as the root is concerned, this is the next stage of the infection thread. After the infection thread, this Rhizobium bacteria start penetrating and the root will start curling now. It started curling. And now what happens? The bacteria started penetrating now. It started penetrating substantially, and now this reaches the cortex region. Cells which reach the cortex, the bacteria which reaches the cortex region, and then it divides, and then it starts forming a vesicles. It starts forming the vesicle, and then it goes to the cortical region, and then the infection thread started propagating. Then from the plasma membrane, from there it starts penetrating and goes inside, and you could see the pericycle, and this region, this started penetrating inside. Now the Rhizobium bacteria through the infection thread which penetrated inside, this started dividing inside in the root cortex region. Initially it was in the thread, now this started penetrating inside and this reached the cortex region, and it forms a peculiar structure. This structure they call it as a bacteroids, the bacteroid structure which forms. So when you look at the bacteroid structure, if you see the plant, the structures, if you could see, this is for example one particular cell I'm talking about. This one particular cell I'm talking about. Here you could see the bacteroid structure. This is all the bacteroids which resemble inside. So this is all the bacteroids. Now the Rhizobium which turns to the structure, this we call it as the bacteroids which present in the cell. And these bacteroids which are surrounded by a layer. This is slender layer that you could see onto which the bacteroids. This is called as leg hemoglobin. This is called as leg hemoglobin, which will be surrounding the bacterial formation. This kind of bacterial structures which form inside, and this is the leg hemoglobin, and this which acts like a scavenger of the oxygen. Because why the reason? Because this is the place where the nitrogenase enzyme is going to get activated. As I already told you, nitrogenase enzymes completely requires absence of oxygen. Which completely requires absence of oxygen. That time what happens? You could see this is actually the bacterial formation. The bacterium, the Rhizobium is there inside, and which is surrounded by the leg hemoglobin. The leg hemoglobin which acts like the scavenger of the oxygen. So the micro anaerobic environment is getting maintained inside the cell. So this is an ideal place by which the nitrogenase enzyme can be activated. Okay. So this where it's happening in the root cortex region, where you could see the bacteroid structures, and this dividing cells with now from the cortex region, it's reaching inside the pericycle. Now it's reaching, so you could see it's reaching in the pericycle of the stem. Okay, it's reaching the pericycle. Now now what happens like this? Once it reaches the pericycle, now the root nodules it started developing now. The root nodules it started developing. Now, substantially what will happen? The nodule vascular tissue which forms completely. So completely the bacteroids which started proliferating inside the pericycle region, this nodule will started developing substantially. So on the whole, this particular zone together we call it as an infected zone. Now, at last, what you see the nodule. This is what we call it as a nodule. This continues to grow into the vascular tissues which reach xylem, right? It goes to the xylem and the phloem, the vascular tissues, and the steel which started developing. And this carries the nitrogenous compounds to the nodule into the steel, and this will be carried to the rest of the plant. This is the place where the nitrogen fixation takes place. This is the place the nitrogen fixation takes place, but the condition where it supposed to maintain is the anaerobic condition. The reason why, because the nitrogenase enzyme which requires absence of oxygen. In the absence of oxygen, it fixes the atmospheric nitrogen. Then, by means of influencing the microenvironment in anaerobic condition, this can be activated. Fine. So this together, the process we call it as a nodule formation. This is the first stage of nitrogen fixation. Once again, I recap what are the stages involved in the root nodule formation. The things that you require, definitely it's a bacterial species supposed to be there in a soil. It was like an asymbiotic mode. Once this started producing the sugars, this bacteria will be greatly influenced towards the root hair. Then it stimulates the infection thread. Through the infection thread, the root hairs curl and then the rhizobium start penetrate inside. Then it reaches the cortex region and it forms a bacteroid structure. The bacteroids are the structures where you could see the bacterial structures inside, which is surrounded by the leg hemoglobin. Leg hemoglobins are the scavengers of the oxygen, and the developing root nodules which takes place and then which leads to the non-vascular tissue which leads, that is, to the vascular tissues which can penetrate the xylem and phloem, and complete nodule formation takes place. Okay, so this leads to the infection. So nodule formation occur and then the nitrogenase enzyme activation takes place. Fine. So this is about the first stage of the nitrogen fixation that we discussed about. This is a nodule formation. So first thing we done. The second part. Coming to the second part, as far as the nitrogen, as I have already told you, like how the nitrogen is supposed to be. So whatever the form which is present in the atmosphere, dinitrogen, this cannot be taken up by the plant directly, right? So it has to be converted to the simpler forms. So it has to be converted into the simpler form. Please take previous tabular column. Which tabular column, Priyanka, you are asking about? The previous tabular column. Which tabular column you're talking about? Let me show the tabular column for you. So this is the only tabular column I've shown you. This is actually the classification of the nitrogen fixation, and this is for the examples, the symbiotic and asymbiotic. I hope this is what the tabular column you're asking for, right? So this tabular column represented free living nitrogen fixing organisms with the symbiotic organism systems. I mentioned the examples, and moreover, like I classified separately. That is like cyanobacteria, that is a free living, and cyanobacteria. And for bacteria also, I categorize the free living and the symbiotic organisms. So this is what, if you're not clear, the sponsor seems to be not clear in this part, definitely I'll send it across in a group so that we can have this for your reference. Okay, so this is the table for the free living and the symbiotic systems. Okay, so coming to the next part. So first we then discuss about the nitrogen fixation, that is root nodule formation. Now we discuss about nitrogenase enzyme. Coming to the nitrogenase enzyme, as we discussed already, the nitrogenase enzyme which requires the microenvironment, that is anaerobic condition. So we discuss about the nitrogenase enzyme, nitrogenase enzyme. Coming to this nitrogenase enzyme, so what the nitrogenase enzyme which consists of ferric, and this consists of the molybdenum. These are the two subunits that you could see in case of nitrogenase enzyme. And this nitrogenase enzyme usually activated in the absence of oxygen. So it requires microenvironment, where you could see at this microenvironment and anaerobic conditions where you could see the nitrogenase enzyme can be activated. So this is a diagrammatic representation which shows like how the nitrogenase enzyme is getting activated. So here, this is the enzyme, the nitrogenase enzyme we're talking about. So this is the nitrogenase enzyme, right? And here you could see the substrate. As far as nitrogen is concerned, the nitrogen is actually, we have two molecules here, and this has been connected with 3 hydrogen bonds. You could see the nitrogens being connected with 3. So 3 hydrogen bonds that you could see. So further to that, what happens? This to the substrate, what happens? The nitrogen is molecules which come and bind, and then what happens? The reduction takes place. As I already told you, these 2 nitrogen, these molecules been bound with the 3 hydrogen bonds. Then by means of the hydrogenase enzyme, what happens? When the hydrogenase enzyme which acts, then what happens? There will be removal of 1 hydrogen bonds. Then 1 H will come and bind over here. Then again the reduction takes place. Then again what will happen? 2 molecules of like hydrogen which comes in bind over here. Then again this reduction takes place. Again the bonds which breaks between reduction takes place between these 2 nitrogen. Then what happens? You could see 3. So here 3 hydrogen with this N, and here it's 3. So eventually what you get the ammonia. So ammonia is NH3. You get 2 NH3 on the whole from 1 nitrogen molecules, and there will be release of products which occurs, and then you could see that the free nitrogen is combined with another molecules of this thing. So this is how the process where the nitrogenase enzymes will fix the atmospheric nitrogen. So how it is happening? By means of the nitrogenase enzyme which is going with the stages. What is the stages here? You could see the nitrogenase, where you could see 2 components, ferric and the ferric molybdenum part. And what are the requirements for this nitrogenase enzyme? As far as the nitrogenase enzyme, the requirements are leg hemoglobin. What are the things it requires for the nitrogenase enzyme? It requires leg hemoglobin, and also it requires adenosine triphosphate. It requires leg hemoglobin. It requires adenosine triphosphate. Apart from this, it requires carbon compounds. This is also required carbon compounds. Also requires carbon compounds. Fine. It requires carbon compounds for what? To bind with ammonia, and also this requires hydrogen donors. Hydrogen donors might be anything. Might be, you could see like pyruvic acid. It may be the sucrose. It may be anything. So you require hydrogen donors, pyruvic acid, or maybe a sucrose. It may be a glucose, whatever it might be. So these are the things which are required for the activation of nitrogenase enzyme. Okay. Absence of oxygen means anaerobic condition. So absence of oxygen means what? It requires presence of carbon dioxide. So the nitrogenase enzyme which requires carbon dioxide, so no oxygen should be there. Okay, for that only it requires what? Leg hemoglobin. What the leg hemoglobin? The main function, it reduces, which acts like a scavengers of oxygen. So that is what the main function of the leg hemoglobin. I told you in the bacterial formation, the bacteria is surrounded by leg hemoglobin. Why it's surrounded by leg hemoglobin? Because the leg hemoglobin which acts like the scavengers of oxygen, so it reduces the oxygen and maintains the anaerobic condition. The microenvironment is maintained for the action of the nitrogenase enzyme, because the nitrogenase enzyme which requires the carbon dioxide, Okay, so and leg hemoglobin, yes, of course, it's a pinkish pigment. Yes, Christian, you are correct. Leg hemoglobin is also the pinkish pigment. If you could see this pigment, it will be a pinkish in color, the leg hemoglobin, and also we call it as a scavenger of RBC. Thank you. So this is the diagrammatic representation of the nitrogenase enzyme. So when you draw the diagram, if you usually draw the diagram of nitrogenase enzyme, how you draw? So you draw the diagram like this. So one here, nitrogen here, and here you could see the other. This is actually the nitrogenase enzyme. This is a nitrogenase enzyme. This is the nitrogen. You could see this is the nitrogen molecule. This is being connected with 3 hydrogen bonds. This is connected with 3 hydrogen bonds. Okay, this is nitrogen. So this is a nitrogenase enzyme onto which you could see the nitrogen. So binding site where you could see the substrate, the nitrogen gas which comes in bind. In between you could see 3 hydrogen bonds which occurs. So what happens, the next stage by means of the hydrogenase enzyme. So by means here you could see the hydrogenase enzyme. This hydrogenase enzyme is responsible for reduction reaction. So what happens? 3 hydrogen bond will become 2. Then 2 will become 1. Then 1 will become null. Then what happens? It will be carried with the 3 hydrogen. Then the product will be formed. Then 1 nitrogen. This molecule is surrounded by 3H over here. So this is NH3. This is how actually the molecules, the product is getting formed, that is ammonia. Ammonia is getting formed by means of the nitrogenase enzyme. Here the that is nitrogenase enzyme by means of hydrogenase. Hydrogenase reduction takes place. In all the cases, the reduction takes place where there is a reduction of the hydrogen bonds which occurs between the two nitrogens. So you could see the three hydrogen bonds you could see by means of the reduction process, where the reduction of the hydrogen bonds, then automatically what will happen? Here you could see three hydrogen bonds which has been connected. So this leads to NH3. So one particular gas, the nitrogen, which gives two ammonia. That is NH3, and NH3 will be formed. Here, pyruvic acid, it may be sucrose. It may be sucrose or it may be glucose. It may be glucose. So this may be a hydrogen donors. So what are the requirements? These are the things I mentioned, the requirements for the nitrogenase. What are the requirements? Leg hemoglobin, ATP, carbon compounds that is required, and then we require hydrogen donors, that pyruvic acid, sucrose, and we require glucose. So these are the things which is required for this one. Okay. So this is actually, it's not a hemoglobin or only red color, but leg hemoglobin is a pink. It's actually the pink one. Okay. And nitrogenase enzyme, which consists of two important units. One is ferric part, other one is the molybdenum part. This type also you get a question. So the nitrogenase enzyme, which consists of two main units. One is the ferric unit and the other one is the molybdenum unit. Okay, fine. This is about the nitrogenase enzyme, because at the time of nitrogen fixation, when you're talking about the root nodal formation, you also should understand about this process. I mean the nitrogenase enzyme, how it performs, what are the requirements for the nitrogenase enzyme that you should understand. So coming to the next part, that is the chemical events which takes place. Chemical events which takes place in the nitrogen fixation. One second, guys. Yeah, what's the doubt, questions? Welcome back. Yeah. Any other hemoglobin is there. So we have prominent, when we talk about the nitrogen fixation, we have this type of hemoglobin, that is leg hemoglobin. We have myoglobin, we have like an oxyhemoglobin, carboxyhemoglobin, these all we discussed, carbaminohemoglobin, these all we discussed, right? These are different types of hemoglobin because it depends upon the carrier, whether it's carrying like oxygen or carbon dioxide or carbon monoxide, this hemoglobin can be classified. Okay, MO protein, yes, MO protein, it's a part of a nitrogenase because it consists of two parts, ferric part and the molybdenum. There are two parts which you could see in the nitrogenase enzyme. Okay, so nitrogen fixation happens in all plants. Yes, of course, the plants should go with the nitrogen fixation. It goes with all plants, but there are few plants where you could see like succulent plants, like where the nitrogen fixation which will not be take place like as we expect, it not by the proportion of nitrogen fixation which is compared, because the environment seems to be not good, where the soil is not having proper organisms like the nutrients and all these factors, then that part like where we cannot expect in all the cases where the nitrogen, but it is common mechanism. Nitrogen fixation is a common mechanism that you could see in all the plants. Sudar right? You cannot understand because you are teaching too fast. I told you very clearly, Sudar because biology is concerned, we have lots of chapters. We have so many chapters. I think you skipped the previous thing, the explanation I gave for this. I do understand I'm going a little bit fast. The reason I told you, because we have a lot of chapters to discuss. We completed only like 10 chapters, I think so, for 11th grade. We have a lot of other more chapters to discuss, and the content in biology is more. Since we have to cover more things, right? So I agree, I'm going a little bit fast. I agree. But if you have any doubts or any clarification, you can ask me. So I'm very happy to explain those doubts for you. So, but I'm in a position to go a little bit fast. I hope you guys can understand, because biology, where we have more chapters, because we have the 11th grade and 12th grade, both we have to complete, and the contents I need to complete on the whole. I cannot ignore any of the subtopics, right? Since I want to teach everything for you. For that only I'm going a little bit fast. So if you have any doubts, so you please ask me. I try to clarify your doubts. So you need not worry about this. Okay. Thank you for understanding. So, but guys, you still think like, I need to go a little bit slow. I can do it, but possibly like, the extensions of, we cannot complete the chapters within the 2 sessions. It may go to 3 or 4 sessions. If it is okay for you, then I will go a little bit slow. I have no issues in going a little bit slow. I can go slow, but thing is the content is more, right? Once I can complete, you can visit the test paper. You spend more of your time for your practice. Okay, you spend more of your time for your practice. That's what my humble suggestion. You can sit for 2 hours, 3 hours also, listen to the class, but only if you practice, you can get good marks. So you practice and practice and practice. If you have any doubts in the subjects or context, because the group they shared my number, you please contact me, and I'm happy to clear all your doubts. Okay, so don't have the thought like I'm going fast. Yes, I'm going fast, but the reason is that the content is more as far as biology is concerned. Okay, thank you. Thanks for understanding. So coming to the next part, we're done with the biological nitrogen fixation. The first part, that is the root nodule. This is the overall the diagram that you could see. You would have studied in the textbook itself. This is about the nitrogen cycle we're talking about. Coming to this nitrogen cycle, we have the animal biomass. This diagram is not new for you. Okay, so how this process, various process is going to take place, that you could see in this diagram. Here you could see ammonification. The ammonification. Then we have a denitrification. Then we have the biological nitrogen fixation. Under the biological nitrogen fixation, we already discussed about the industrial nitrogen fixation and also electrical nitrogen fixation. There you could see the conversion of like ammonia to nitrate and nitrate, that is denitrification process, right? Nitrification process. So nitrification, so nitrification, denitrification, assimilation of nitrate, then fate of ammonia, all these things that we're going to discuss like substantially. So this is just an overview how the nitrogen cycles, how the atmospheric nitrogen. This is atmospheric nitrogen, we call it as dinitrogen. This we call it as dinitrogen. So this getting fixed either by like industrial nitrogen fixation, then electrical nitrogen fixation. Then the process of nitrification, like nitrate, which is getting ammonia to nitrate, and nitrate is getting converted to nitrate, right? This is the nitrification. And then how this converted forms like ammonia, which is again taken and replaced by the plants, that we discuss as an assimilation, that we discuss separately as assimilation. Then the denitrification by the dead and decaying organisms, like how the denitrification process. So together, as far as nitrogen cycle is concerned, ammonification, nitrification, denitrification. These are the important areas that we're going to cover, and also we discussed about assimilation. Okay, so coming to the next part, the chemical events in the nitrogen fixation. The first we discussed about the nodule formation. Next we discussed about the chemical events, in nitrogen fixation. So what are the things that requires for this, the chemical events for nitrogen fixation? It's reducing power. It's reducing power. Reducing power, you could see Na, DPH, FMNH2. Can someone give the expansion for NADPH? What is the expansion for NADPH? And what is the expansion for FMNH2? What is the expansion for this two? So electrical nitrogen fixations, others two are clear for me. I told you electrical nitrogen fixation by means of electric discharges. We discussed in the lightning, thundering that we discussed, right? Electrical mode of discharge. In the beginning part, I hope you discussed about like a nitrous oxide, nitrogen peroxide. These are the areas that ozonization. These are the areas that we discussed, right? So there we discussed about electrical discharge. Okay, then soil nitrogen pool. This is the soil nitrogen pool is actually the combination of the nitrification process where ammonia is getting converted to nitrite and nitrite is converted to nitrate. So nitrogen pool, this we call it as nitrification, where this is ammonia. Ammonia is getting converted to nitrite. This is getting converted to nitrite. First is nitrite comes, nitrite, and then it is getting converted to nitrate. Right? This together mechanisms which occurs together, we call it as a soil nitrogen pool. This we call it as nitrification. Okay, so this process, which process you're talking about? Like what is this process? This complete process we call it as nitrogen fixation. The nitrogen fixation, I mean the symbiotic and asymbiotic mode of nitrogen fixation. where we have the categorization. We have categorization, ammonification, nitrification, and we have a denitrification. Clear? Now we move to the next part, that is the chemical events in a nitrogen fixation, which requires the reducing power, that is NADPH and FMNH2, and this also requires energy. That is ATP. This also requires energy. ATP it requires. Then it requires dinitrogenase, and this requires carbon component. For what? For trapping ammonia. So these are all the chemical events which takes place in the nitrogen fixation. So when I represent the overall reaction for this, how can I mention the nitrogen plus 8 electrons plus 8 protons plus you take 16 ATP, right? The 16 ATP which gives rise to 2 molecules of ammonia. So 2 molecules of ammonia which is getting formed plus 2H plus 16 ADP plus phosphate ions. This is how actually the overall reactions. Yes, nitrification involves 2 stages, nitrate and nitrate. 2 stages that you could see in case of nitrification. I will explain the bacteria. There are specific bacteria which is involved for nitrate formation, and we have some specific group of bacteria for the nitrate. So coming to the chemical events that you could see, this process is called as nitrogen fixation. The first we discussed about root nodule formation, and then this is the nitrogen fixation. For the nitrogen fixation, what are the things that is required? It requires energy, dinitrogenase enzyme, carbon compound for trapping ammonia, and if you could see the overall reaction that you could see is nitrogen plus 8 electrons plus 8 protons plus 6 ATP, which gives rise to 2 molecules of ammonia plus 2 H and plus 16 ADP and the 16 phosphate. On the whole, this equation represents nitrogen fixation. Sometimes they give this equation and they'll ask you to what type of reaction, and sometimes they give the reaction for denitrification. So in this part, you also should be very careful with the equation. This overall reaction represents the nitrogen fixation. So coming to the biological nitrogen fixation, first we discussed about root nodule formation. Second we discussed about the nitrogen fixation. Okay, guys, we're clear with this part, the nitrogen fixation. Next, we move on to the third stage, next to the nitrification, that is nitrogen fixation. We'll start with nitrification. Coming to the nitrification, so as I told you, the nitrification which combines, which is of the ammonia which is getting converted to nitrate, and then it converted to nitrate. Fine. So coming to the next part, that is the third part. So root nodule formation we have done. The second we done is nitrogen fixation. The third part is we discussed about nitrification. Coming to nitrification. So what is nitrification? So plant absorb ammonium ions. So plant absorb the ammonium ions and is more adapted to nitrate. So what is happening here in nitrification? There is a conversion of ammonia to nitrate. This process together we call it as a nitrification. So conversion of ammonia to nitrate, and this nitrate is actually which used by the plants. So this nitrate only is going to be used by the plants, because the plants which cannot take up directly. So the ammonia has to be converted to nitrate. This nitrate are ammonium ions which can be taken by the plants actually. Okay. Equation again you are asking for. Yes, the equation is N2 plus 8 electrons plus 8 protons plus 16 ATP, which gives rise to 2NH3 plus 2H plus 16 ADP plus 16 phosphate. Okay, this is overall reaction for the nitrogen fixation. Clear? Okay, now we move on to the nitrification process. So nitrification process is the conversion of ammonia. Ammonia is going to be converted to nitrate. So how this process takes place? This process takes place by means of 2 things. First thing is the ammonia has to be converted to nitrate, and then it has to be converted to nitrate. First TRITE nitrate, then A. Nitrite first and then comes the nitrate. Okay. So usually the bacteria which are responsible for this type of nitrification process is chemoautotrophic bacteria. So usually chemoautotrophic bacteria which plays a very important role for this process, that is nitrification. So NO2 for nitrate, and NO3 can be represented as NO3. So what happens in case of nitrate? So the reaction is ammonia. So NH3 to NH3, which combines the 3 molecules of oxygen, which gives rise to 2NO2 plus 2H plus 2H2O. This gives rise to minus 84 kilocalories of energy. The energy which is like liberated out of this nitrate is minus 84 kilocalories. This is the overall reaction that you could see in case of nitrate formation, where the ammonia is getting converted to nitrate. The ammonia is getting converted to nitrate. So 2NH3 plus 3O2, which gives rise to 2NO2 plus 2H plus 2H2O. The organisms which are responsible for first process of nitrification, it is Nitrosomonas. This is very important. Nitrosomonas, the bacteria. This you should remember. How the ammonia is getting converted to nitrate? This is by means of Nitrosomonas. Coming to this now, this nitrate has been formed. Nitrate has to be converted to nitrate. How this can be converted to NO2, which combines with the oxygen, right? Now this nitrate which reacts with the oxygen and this converted to nitrate, this get converted into NO3. Can you tell me what is the name of the organism which is responsible for nitrate? As far as nitrate is concerned, Nitrobacter, Nitrocystis, Nitrobacter. Guys, this area is very important. And Nitrocystis. Mostly the questions which comes, which of the organisms responsible for converting ammonia to nitrate is Nitrosomonas, and nitrite is getting converted to nitrate. This by means of Nitrobacter, and this gives the energy of minus 17.8. It gives minus 17.8 kilocalories of energy. This is for the nitrate formation. Together, nitrate, ammonia is getting converted to nitrate, and eventually the nitrate is getting converted to nitrate, right? This together we call it as a nitrification, where the conversion of ammonia to nitrate. But there are few organisms. For one organism that you could see, the fungi, the mold, which performs both the reaction, which performs both nitrate, that means it converts to nitrate as also which can be converted to nitrate. Can you name the fungi? Can you name the one mold is there? There is one mold, the fungi. Can you name the mold which performs both the function? It means it can convert to ammonia to nitrate, and also this can convert nitrate to nitrate. But more specifically, we say this mechanism Nitrosomonas, nitrite to nitrate, it is Nitrobacter and Nitrocystis. Fine. But there is one particular mold that you could see which performs both, which you could see in both the cases. What is the name of the fungus? What is the name of the fungus? I mean, what is the name of the mold that you could call it? As we start with A. What is the name of the fungus? What is the name of the fungus which performs both, that is nitrate, which converts to nitrate, and also the nitrate also be converted to nitrate? Can someone tell me what is the name of the fungi, that is a mold which could see in both the reactions? Okay, so this area you should be very careful. In this area you get the questions from either from Nitrosomonas and Nitrobacter. Just one word is different. The first which comes is I, nitrite. Ammonia is getting converted to nitrite. Then next comes A, nitrate. So first is nitrate, and then when it comes nitrate. In this case it is Nitrosomonas, and second case I could see Nitrobacter and Nitrocystis. It is Nitrocystis. Okay, so the name of the mold is Aspergillus flavus. This you could see in both the cases wherein nitrite as well as that you could see in case of nitrate. Okay, so this is the process called as nitrification, where ammonia is getting converted to nitrate, and the nitrate is also eventually is getting converted to nitrate. Next we move on to ammonification. We move to the next part, ammonification. Coming to ammonification, let me clear this. So ammonification is the fourth part. Ammonification. So we done the nodule formation, nitrogen fixation. Then we discussed nitrification we discussed, and more specifically aspergillus. We have different species of aspergillus. We have aspergillus niger, different species. But aspergillus flavus is very specific that you could see. Okay. Next comes to ammonification. Coming to ammonifications, what is going to happen in this case of ammonification? Where any organic matter, okay, or nitrogen containing compound, or any nitrogen containing compound, which is getting converted to ammonia. This process we call it as ammonification. Any organic matter or any nitrogen containing compound which is getting converted to ammonia. So the formation of ammonia is usually we call it as ammonification. This usually performed by decomposition. Of organisms. Decomposition of most of the organisms. Decomposition by the microorganisms. These microorganisms are dead organisms which decompose. Example you could see in this case where usually Bacillus species. Bacillus racemosus. This is a bacteria, Bacillus racemosus, which involves in the ammonification process, and Bacillus vulgaris, Clostridium, Bacillus vulgaris, then Clostridium. This can also perform this particular function, that is conversion of organic matter to ammonia. Also, you take actinomycetes, even the members of actinomycetes, which that you also you can see in this particular process. So what's happening in this case ammonification, where the protein is getting converted to ammonia. Protein is getting converted to amino acid with the help of water molecule. Then again what happens is amino acid which will be keep on again getting converted with the water molecule, it converted to organic acid, and this leads to the formation of ammonia. So this process which we call it as ammonification, here the nitrogen changed from the gaseous to the ionic form in the soil, where the pH should be more than 6. The pH should be more than 6. Where you could see in certain exception you could see in Oxalis begonia. In these cases where this can fix the ammonification process at the pH of 6. But in this case, the remaining majority of the cases, what happens? The nitrogen will change from the gaseous to the ionic form in the soil at the pH of more than 6. pH usually it is more than 6 in the normal ammonification process. But there are exceptions where this process ammonification which takes place even lesser than 7. That is in Oxalis and begonia. These plants where you could see this ammonification slightly acidic, we could see at the pH of 6 that it takes place. But usually the ammonification which takes place, it means the nitrogenase, that is the ammonia, which change from the gaseous to the ionic form in the soil. That is like the pH that is more than 6. Okay, this is about the ammonification process. And there are certain places where you could see the same process which can occur in the non-leguminous plants also. This is the usual process we're talking about ammonification, nitrifications and everything which is happening. But when you take in case of like asymbiotic, that is in the non-leguminous plants. If you take in non-leguminous plants, the same sort of like the process which takes place, ammonification, all this process takes place. Say like an example in take of Frankia, which consists of lipid compounds. And this lipid compounds which has a specialized structure that you could see, this we call it as an oponoids. Can you tell me what is the function of oponoids? This in non-leguminous plants, especially in case of example you take in Frankia. In Frankia, this consists of lipid compounds like oponoids. Okay. In oponoids, what is actually the function? The oponoids which forms like surrounded by, which is like vesicle, and this we call it as a diazo vesicle. This oponoids which forms a diazo vesicle. So can you tell me what is the function of diazovesicles which form in case of non-leguminous plants? This is the normal process which occurs in leguminous plants. There are non-leguminous plants like Frankia, the Actinomycetes family, Actinomycetes, where you could see this type of lipid compounds which consist of openoids. The openoids which forms a vesicle-like structure. This vesicles we call it as diazovesicle. What is the main functions of this diazovesicle? Cancer is very that we already discussed. What is the main function of this openoids, or what is the main function of this diazovesicle in case of non-leguminous plants? We discussed some about the leguminous plants at the time of root nodal formation, but in non-leguminous plants, we have the structure called as openoids here. So what might be the function of this openoids in case of non-leguminous plants? Because the non-leguminous plants may be not having a symbiotic relationship. So what might be the case in case of function of this openoid case? Okay, how will they ask questions from this? From this, they'll ask questions usually with the microorganisms. They ask a question. Okay, they ask questions from the microorganisms. Ammonification. What is it? Find out the name of the bacteria which are responsible for ammonification. Or they'll give an equation. They'll ask you to identify. Find out the bacteria. From ammonia to nitrate, they'll ask you to identify the name of the bacteria. Or from nitrite to nitrate, they'll give an equation. They'll ask you to identify the nitrate reducing organism. So you need to remember the name of the microorganisms. So name of the microorganisms very important in this particular subtopics where ammonification, nitrification, you take denitrifications, any process that you take, the name of the organisms is very important, and also you should remember what are all symbiotic, what are all asymbiotic. This is the important area that you can get a question from this part. Okay, and also the legumoglobin, nitrogenase enzyme. So this is one important part that you should be very careful with. So we're done with the ammonification process. Move on to the next part of ammonification, nitrogen assimilation. Coming to the nitrogen assimilation, how the nitrogen can be assimilated. So now the nitrate has been given. So this has to be taken by the plants. So how it can be taken by the plants. Now we'll discuss this process. So nitrogen assimilation. How it's going to, now it's ready for the plants. Now it's up to the plants how it's going to absorb or how this is going to get taken up. Now the microorganisms perform the function. Now the plants has to reduce the nitrate to ammonia. So now what the plants, because the nitrate has been formed by means of nitrification. Now the plants has to again convert this nitrate. This has to be converted to ammonia. This should be converted to ammonia. First, what should happen here in this case? Nitrate has to be converted to nitrate. So first case, what's going to happen is nitrate has to be converted to nitrite. The second thing is nitrite is again going to convert it to ammonia. It's going to convert it to ammonia. So this we call it as nitrogen assimilation. This part is called as nitrate reduction, and this nitrate reduction, this one is nitrate reduction. First, nitrate is getting converted to nitrate. Where this process is going to occur? This occurs in the cytoplasm, that is in cytosol, which occurs. Nitrite to ammonia. So this usually occurs in the chloroplast. It occurs in the chloroplast of leaf cells. It usually occurs in the chloroplast of the leaf cells. So nitrate is getting converted to nitrate. So what's the reaction that you could see? Here is NO3 plus NADPH plus H is getting converted to nitrate. That is NO2 plus H2O plus NADP. Can you tell me what is the enzyme? Sometimes they'll ask you the question, what is the enzyme? What is the enzyme which converts nitrate to nitrate? So here the enzyme is nitrate reductase. The enzyme here is nitrate. They ask question like this. They ask reductase. They just mark empty here. X or something they mention. They ask what does the X represents. They are asking for enzyme. So the enzyme which is responsible for converting nitrate to nitrite is nitrate reductase, where nitrate is converted to nitrate. Okay. So this usually nitrate reductase is a molybdenum flavoprotein. This is a, what's the component? It is a molybdenum flavoprotein. This enzyme, it's a molybdenum flavoprotein. Next, coming to nitrate. This nitrite has to be converted to ammonia. Then what should happen here? 2NO2 plus 7 NADPH plus 7H. This getting converted to 2 NH3 plus H2O, and then since like 7 NADPH here, like you could see 7 NADP, so oxygen. So here the nitrite is getting converted to ammonia. So what's the name of the enzyme? Here the enzyme again the same type, nitrate reductase. Here it is nitrate, here it is nitrite reductase is the enzyme. So this is how like an assimilation of the nitrogen takes place. So nitrate reductase in this place, here it surely the ferric and you could see the copper protein. This made up of ferric and copper protein. This also they make an ask a question. In this case, nitrate reductase is a molybdenum flavor protein, but nitrate reductase doesn't have molybdenum. This also makes the difference here. Nitrate, which has got molybdenum flavor protein, but nitrite reductase, which is having only the ferric and the copper. So nitrate reductase followed by the nitrate reductase. In this area, they give importance for the enzyme and ask the difference in what way it may be differentiated. I find out the correct statement. So nitrate reductase, which carries like molybdenum and the flavor protein, nitrite reductase, which carries like ferric and the copper part. This makes the difference. This makes the difference that between that is a nitrogen assimilation is concerned. So this nitrate reduction and the both these reactions, the importance is what where it occurs actually. It occurs very fast in the light source. If the light is present, this both the reaction which occurs faster in the light. Okay, so it occurs faster in the light. This reaction. So if the light source is comparatively less, then again the assimilation of ammonia is also getting, because it's one of the factor. The light is one of the important factor which facilitates the reaction. Okay, so we're done with this part. The last part we're going to discuss is the fate of ammonia. Coming to the fate of ammonia, now the ammonia has been formed. So what is the fate of ammonia? The fate of ammonia where ammonia should go to this process. Right. So what is the process where then ammonia? You could see NH3 has been formed, right? Now the NH3 has to be converted to NH4. NH4 plus alpha keto glutaric acid. It forms alpha keto glutaric acid plus NADPH. NADPH. This produces what? This produces amino acid. What amino acid here? Glutamate or glutamic acid. This forms glutamic acid. Okay. So this glutamic acid is formed. So the process of this is called it as fate of ammonia. This slide shows what will be the fate of ammonia. The fate of ammonia. What will happen here in this case? First, the reductive amination which takes place. Reductive amination takes place. In case of reductive aminations, where the ammonia is getting converted to ammonium ions with the alpha ketoglutarate, right? With the help of enzyme. What is the name of the enzyme here? Glutamate dehydrogenase. This enzyme. By means of this enzyme, that is glutamate dehydrogenase with the presence of NADP, then it be converted to glutamate, that is glutamic acid. So this is how actually the amino acid is getting formed. Then what happens, like this glutamic acid which produce other different types of amino acids. So first amino acid which is produced is glutamic acid by means of reductive amination. This process, what's the product that you receive? You receive this glutamic acid, this particular amino acid that we receive. From this glutamic acid, the other type of amino acids which also started producing. So other amino acids are synthesized, and eventually what left, this also leads to the amide formation. Second process, what is transamination? The transfer of amino group which takes place. This is also like fate of ammonia, the transamination. This is the amino acids which is getting converted to alpha keto acid. Then you take alpha keto acids, the transfer of the amino groups which takes place by means of the enzyme which call it as transaminase enzyme. This also which will be taking place. But sometimes in case of soybean, instead of ammonia is getting assimilated, the fate of ammonia, where in case of soybean what you could see is urids which is getting formed. Can you someone tell me what you mean by urids? What do you mean by urids? Especially in this type of plant, the soya bean, you could see the urids are getting formed. So these urids are getting formed. This is actually like, where what is happening in case of the urids, where the nitrogen is getting translocated. Urids is something where you could see the nitrogen is translocated. Instead of glutamic acid, here the nitrogen is getting translocated, right? So the fate of ammonia is concerned, it can occur by means of reductive amination, or the transfer of amino groups by means of the transamination that can happen, or catalytic amination you take. Catalytic amination, what happens like ammonia which combines with, so what happens with catalytic amination? The ammonia which is being formed, this ammonia which combines with what? Which combines with amino acid, and this form amines, which form amine. This is called catalytic amination. So these are the three ways by which we call it as a fate of ammonia. First, by means of reductive amination, where you could see the ammonia is getting converted to glutamic acid. From the glutamic acid, the other amino acids are getting formed. The second process is by means of transamination. So where the transfer of amino groups which is getting formed. Then the third type is catalytic. This is the third type, the catalytic amination. The ammonia which combines with the amino acid, where the amide is going to get formed here. So amides are what? Amides are carrying more amount of nitrogen. So it's having more amount of nitrogen. Amides usually carrying a more amount of nitrogen. Now this what will happen? This will be translocated. This will be translocated to what? Translocated to xylem. The vascular tissue. This will be translocated to xylem. So, but in case of like certain cases like soya bean, where you could see the urids instead of amides, like it will be directly the urids will be formed where the nitrogen itself is getting translocated. Say like an example, soya bean. So this is the fate of ammonia. So three steps that you should remember. Reductive amination, you should remember the glutamic acid that you should remember. From this, the other amino acids are formed. Transamination, fine. The third thing is catalytic amination, where the ammonia is combined with the amino acid. Okay, fine. So this is about the fate of ammonia. Now we move to the next part, the last part of this chapter, that is denitrification. So you might have studied the denitrification process. Coming to denitrification, the last process that we discuss is denitrification. Denitrification. Fine. It's a denitrification. So, what is the denitrification? Some microorganisms use nitrate. So, some microorganisms, MO is microorganism. Microorganisms use nitrate. Okay. So, some microorganisms use this type of nitrate under anaerobic conditions. So, usually used under absence of oxygen, that is anaerobic. Can you give some example? Can you give some example for denitrification? So guys, you need to be very careful with the nitrogen fixation, the organisms, ammonification, then we have the nitrification, the organisms, then also for the denitrification. So yeah, we have class till at 4 o'clock. We have till at 4 because we have begun with 2 o'clock, no? 2 o'clock we begin the session. Okay. Yeah, we have class till at 4 because we began the session at 2 o'clock. We have class till at 4 today. So some microorganisms use nitrate under the anaerobic conditions. Example. Can I give an example? Usually of Pseudomonas. It's a bacteria. Pseudomonas denitrificans. They give a different species. Pseudomonas aeruginosa, Pseudomonas flavus, the different organisms. Here, this is by means of Pseudomonas denitrificans and by also thiodenitrificans. Thio means thiobacillus. Thiobacillus we call it as thiodenitrificans. And also by means of micrococcus. And by means of bacillus. These are some of the organisms which converts the nitrate, which use the nitrate under anaerobic conditions. What the reaction for this? This is like 2NO3, which is getting converted to 2NO2, and this 2 becomes NO, then N2O. Then eventually, what is the product that you're getting is finally N2. So this is how the denitrification process. It substantially takes place for NO3 to NO2. This is NO2, then 2NO, then N2O, then it forms N2. What happens? So if this reaction takes place, then definitely acidity of the soil, the pH of the soil will be greatly affected. So what will become now? The soil will become alkaline or neutral or acidic if this process takes place. Guys, we just look into this reaction. If this reaction takes place, this denitrification, if occurs, right? This, what will be the fate of the soil as far as pH is concerned? Whether the pH will become acidic or pH will become alkaline or pH is neutral. So what will be the fate of the pH of the soil? And also you take achromobacter. Yes, achromobacter. Thio is thiobacillus. No, it's a thio. I just mentioned the short form. Thiobacillus. Thiobacillus denitrificans and also achromobacter. You can take achromobacter. Fine, that is correct. So, what is the pH? Now, definitely the pH of the soil is not good sometimes where the pH will become acidic. So, if this denitrification process, that means where the microorganisms use the nitrate under anaerobic conditions, definitely the soil will become, it causes acidification of the soil. So, we done with this part, the denitrification, nitrification, all the areas that we have covered. The last stage of this particular chapter, there is other mode of nutrition also. We come to the last mode of nutrition. We're almost done with the nitrogen cycle. The areas like ammonification, nitrification, the nitrogen fixation, denitrification, then nitrogen nitrate assimilation the nitrogen assimilation we discussed, then fate of ammonia that we discussed. Guys, in this area, you should be very careful with the type of microorganisms, especially the symbiotic. You should be very careful with the type of organisms, that you should be very careful in this particular area. Okay, fine. So last part is heterotrophic mode of nutrition. So heterotrophic mode of nutrition that we'll discuss. Coming to this part, the last part of this chapter, heterotrophic mode of nutrition. So, coming to this part, heterotrophic mode of nutrition. This of three types actually. Parasitic plants, saprophytic. We're going to give examples for this. Saprophytic, and we have the insectivorous. Insectivorous. Coming to the parasitic, this is the heterotrophic mode of nutrition, right? Parasitic, you take examples. Can you give examples for parasitic plants? Few examples for parasitic plants. Coming to saprophytic plants, example take monotropha. Example, it feed and decaying substance, monotropha. You know the insectivorous plant. You know, can you give some examples for insectivorous? As for insectivorous plant is actually autotrophic and it requires amino acids. So it is autotrophic and it requires amino acids. These amino acids of animal origin. This requires amino acids which must be of animal origin. Fine. Coming to the parasitic mode, cuscutter, then Rafflesia, then Xanthomonas. These are some of the parasitic plants that we could see. Cuscutter, Rafflesia, Xanthomonas. These are the parasitic plants which undergoes heterotrophic mode of nutrition. Saprophytic plants you take like monotropha. Come to the insectivorous plant. Insectivorous plants that you will study like Utricularia, Nepenthes, Utricularia, Nepenthes. These are all the insectivorous, the plants that usually you will study the lower classes, right? Where you could see like which one we call it as an Indian pipe. Can you tell me like which we call it as an Indian pipe? Which plant often we call it as an Indian pipe? We call it as an Indian pipe is actually the common name that we'll give. Which type of plant we call it as an Indian? I given examples, right? Out of these examples, which plant we call it as an Indian pipe? And as the insectivorous plants, which is not completely depends on the soil for the nitrogen, right? So it's not depends on the nitrogen. So what is actually depends because it feed on the insects, right? So for that itself it takes all the necessary the nitrogen. So that is the reason why you could see the insectivorous plants which is not required on the soil. So this can grow in a nitrogen deficient soil. So that is the reason these plants can grow in a nitrogen deficient soil. Why the reason? Why the reason is these insectivorous plants, like which not depend on the soil for the nitrogen, not depend on the nitrogen. So it's actually it's taking from the insects itself, right? So it can grow these plants, which can grow in the nitrogen deficient soil. So Indian pipe is a common term that we give for a monotropha. Monotropha commonly we call it as an Indian pipe. So by this we come to almost the end of this chapter. So that is protein metabolism. Protein metabolism. This is Venus flytrap. These are all the common names that we give for Utricularia, Nepenthes that you study in the lower classes. So these three types of plants which undergoes heterotrophic mode of nutrition. Okay, so definitely I'll send this picture. If you request for the picture, definitely I'll send it across for you. Okay, so the question comes like, enzyme nitrogenase is a what type of protein? What is the unit which is present in the enzyme nitrogenase? So the question comes like this. How do you answer? What type of the complex that you could see in the enzyme nitrogenase? The first question, if I'm asking you, the enzyme nitrogenase. Enzyme nitrogenase. What enzyme nitrogenase which consists of? Can you answer for this question? Enzyme nitrogenase. What type of protein it consists of? Then can you tell me, like, during symbiotic nitrogen fixation, how many ATPs are utilized during symbiotic nitrogen fixation? How many ATPs are utilized? Can someone tell me for this question? How many ATPs are utilized in symbiotic nitrogen fixation? Symbiotic nitrogen fixation. How many ATPs? If this question comes, they have given option. How many ATPs are utilized during the symbiotic nitrogen fixation? So what is the unit that you could see in the enzyme nitrogenase? Bacteria which reduces nitrates in soil. Can you name the bacteria? The bacteria which reduces the nitrates in soil to nitrogen. Nitrate in soil to nitrogen. So what is the name of the bacteria? What is the name of the bacteria? So you ready with something that give the entire equation itself, maybe the nitrogen fixation or the denitrification, the nitrate or nitrate, any particular segment, they'll ask you, they give the equation, they'll ask you to find out the name of the organisms. What is the element? Is a constituent of biotin. Sometimes they ask about element that is a constituent of biotin. Can you name the element which is a constituent of biotin? What is the name of the element which is a constituent of biotin? So what is the enzyme? Because I told you the nitrogenase which consists of two important units. What are the units? How many ATPs that is required? Then what are the bacteria which reduce nitrate to nitrogen? And you could see, like, case like, dieback citrus. You could see the disease name is dieback citrus. And reclamation of legumes. This is the name of the disease. Which element, the deficiency of which element, which cause this one? The dieback citrus. Sometimes they'll ask the dieback citrus and reclamation of legumes. This happens due to the deficiency of what? They'll ask this type of question. For example, if you go for this enzyme nitrogenase, it's like molybdenum ferric protein. It consists of molybdenum ferric protein. Then how many ATPs? You gave the correct answer. It is 16 ATP. 16 ATP is required for fixing this nitrogen fixation, symbiotic nitrogen fixation. Bacteria reduces nitrate to the nitrogen. Then it is, what's the name of the bacteria? It is Pseudomonas. Usually ask questions like this, Pseudomonas. Then elements where you could see the constituent, which is the element where you could say constituent of biotin. Can you name the constituent of biotin? Can someone tell me? And dieback citrus and reclamation of this legumes. This is actually the name of the disease. This caused by the deficiency of what? What is the name of the element? You just tell what's the name of the element which is responsible. Okay, Donan's equilibrium occurs in which surface of the cell? We discussed in last class. Donan's equilibrium. Where it occurs? Which part of the cell? Equilibrium. Occurs in Donan's equilibrium occurs in which part of the cell? So this is because of copper. Then element I told you the constituents of the biotin. So the constituents of biotin, Can someone tell me like what's the constituent of biotin? This is sulfur. This is sulfur. Dominance equilibrium, where the dominance equilibrium which occurs. Dominance equilibrium occurs in which part of the cell? Okay. They ask like in reductive deamination, what is the product which obtains? Reductive deamination. They ask the question, what is the product which is getting formed in a reductive deamination? In reductive deamination, what is the name of the product? What is the name of the product that you could see the reductive deamination? So these are the various type of the questions that you can get. Okay. Okay. So I answer for all the questions like reductive deamination. So reductive deamination, what is the name of the amino acid? It's a glutamate or glutamic acid. Glutamate. It is glutamate. It is glutamate. Okay. Fine. Okay. So dominance equilibrium. Dominance equilibrium which occurs in a plasma membrane. Cell where you could see which occurs in the plasma membrane. So like this, in this particular chapter, they ask questions like whether the microorganisms, the name of the microorganisms, either for ammonification, denitrification, assimilation of ammonia. So be prepared with this type of questions, and I hope like we're done with the chapter today. Tomorrow we'll start with the plant hormones, that is plant growth and development, in which we'll see about the different hormones, the growth and development of the plants like seed dormancy, auxins, gibberellins, cytokinins, all those things that we'll discuss. Okay, so we're done with this chapter. So today on the whole we discussed about the nitrogen fixation, I mean nitrogen metabolism, where the nitrogen fixation, ammonification, nitrification, denitrification, assimilation of the nitrogen and the fate of ammonia, and mainly this part, be very careful with the microorganisms. The last answer, which answer are we talking about? Sixth, yeah. The reductive deamination. What is the product of the reductive deamination? Is glutamate, or you can say glutamic acid, glutamate or glutamic acid. Okay, so I hope like you receive the test papers. You just go through with the test papers, and hopefully like tomorrow we'll see and we'll start with the fresh chapter. Okay, I hope you listen to the session, because I didn't give you a break also today, so we didn't have a break in between. So I just completing just like 5 minutes ahead just before itself. So just go and revise yourself. Thank you so much. Thank you.